Thank you, Marty. So at Thrive, we're providing data about live cells that has not been available. There simply are not the comparable types of data sets about live cells that allow you to do machine learning, data mining. This is my 19th company. I've had 7 unicorns. 5 of them were AI companies, including your credit cards are being monitored by neural networks from a company I started that Isaac bought. Your FICO scores are being generated by neural networks. Pap smears, 90% of all pap smears are being read using neural networks from a company I started, Cytic, which Hologic bought for $6 billion. There's a lot more AI that's out there, but without good data, you're not going to have good machine learning or AI. So our first step is to provide the kind of data sets that are needed for AI. So we provide data, imaging, analytics, and automation. And the applications are basically cell-based experiments and delivery of cell-based therapies. So that includes biomedical research, drug discovery, machine learning, and delivery of medicine or bioprocessing. So our mission is to vastly improve the fundamental tools of studying cells. And cells are the fundamental unit of life. By replacing microscopes and extending them with advanced capabilities, we allow researchers, and we hear this constantly, to see things they couldn't see before. We illuminate the invisible, and we make the complex accessible. Our instruments are replacing the ubiquitous microscope, which should not be being used. This is an analog instrument. It's been around since the late 1800s, and very little of that data is ever captured other than some scribblings in a lab notebook. Yet it is ubiquitous. But we have found that our customers replace their microscopes with our imaging instrument. So they actually kind of led us to that. Unfortunately, we have a lot of breakthrough technologies in regenerative medicine, CAR-T, and others that are really being held back by the lack of data and analytics and automation with live cells. So our products really empower mostly researchers. It accelerates their discovery. It makes their data reproducible. It really allows them to go where they weren't able before. At a practical level, these are some of the applications of our imaging instrument. So stem cells, drug screening, drug response, in vitro fertilization. And by the way, we are often surprised by what our customers do with our instruments. We actually didn't know that we could do organoids and in vitro fertilization. Customers, it's too bad that companies before they start can't have customers. You can certainly try to collaborate and interview them, but there's just nothing like having customers. We've learned so much. We're replacing assays such as a viral plaque assay. Imaging organoids is probably where we contribute the most in the short term, because that is now encouraged by the FDA, and it's the fastest growing area of cell culture. You no longer need to have animal data to go into humans for the FDA, as long as you have good cell culture and tissue culture data. We can image suspension cells, and we can even track something everybody wanted to do and never thought could be done, which is we can track a single cell over months. So better that it comes from a customer than me. One of our customers, I think, did a good summary of the status of where things are now. Current tools are failing us because they hurt the cells, only give us 2D, single point in time images, and don't capture contextual data, such as documentation or enough data. And after using our instrument, he said, Thrive will change the world. Thrive allows us to understand and see living dynamic cells and tissues in 3D over time with remarkable images. Biology takes place over time. It's a process, and it's not flat. It has depth. Cells have depth. And we take images in time series and at 100 focal planes so that you can see what happens to the cells. So we started Thrive because of the reproducibility crisis. I read an article about Amgen that was really pretty incredible in 2013. They tried to reproduce 53 landmark peer-reviewed journal articles, and they could only reproduce 6 of them. Bayer also had a similar study. So basically, the way cell culture is being done is completely manual. The data is not comparable. There's mislabeling rates of up to 31%. This is an FDA survey. And researchers are spending hours per day looking at cells, and then when they put them back in the incubator, all the data is lost. Researchers spend much time trying to reproduce an experiment a second time, but often it's 2 flukes, 2 not real experiments, because there is so much variability. And the amazing thing is, we know so little about these cells, and then we take them and we spend billions on various instrumentation studying them. So cell culture is one of the weakest links, and yet it's the first link in the chain of drug development. And that's really been our vision, is to try and improve the odds of successful drug development. Sadly, this is what a cell culture lab looks like. Uber knows more about us than we do about the cells that we're studying for biomedical research. We have these non-smart incubators. You put your cells in the dark in an incubator. Your cells are in the dark, and so are you. There's no data being collected like we do at Thrive. We're imaging our cells on various schedules that are set up by the researcher. There's really just a dearth of data, and we often hear things like, you enable an experiment we couldn't do before. Center for Genomic Medicine at Massachusetts General Hospital said, for the first time, we have high quality, consistent data to share with collaborators, which is important because right now, data is not consistent. The way it's done collected in one lab is different from others. To vastly improve experiments and to enable machine learning, data mining, and breakthrough insights. So what's wrong with microscopes? Well, the problem is that they're being used sporadically to look at living cells. And computers should be continuously monitoring cells. So microscopy is analog. It's not reproducible. It's inefficient. Researchers spend hours per day with our instrument, minutes per day. They insert the cell culture plate, and in minutes we take 10, 30 gigabytes of images and put them in a database. We're not collecting sufficient data. We take images of what we think is interesting, but that's biased data. That's not the basis for machine learning. Neural networks need all the data they can get, and they'll tell you what's important. You can't bias the data ahead of time. It's not comparable because it's cell culture, and cell culture imaging is done manually, whereas we do it automatically. It's static. Most instruments only take an image at one point in time. If they take it at multiple times, you can't find that same cell again as you can with us. Microscopes are a single focal plane. We're 100 focal planes. Things are going unrecorded in lab notebooks or inaccessible. We record everything. We have a barcode system that's integrated, and we know where the plate is throughout its history. The data is also isolated. All of our instruments are networked and are accessible outside the lab. In a sense, they're global, not local. So just a little background on Thrive. We have 22 employees, 14 in R&D, 13 are actually software and machine learning. We only have one hardware person. We've sold 27 instruments. That's about 5 million in revenues, and we're projecting 128 million in 5 years. Our principal, by far, most common application is drug screening, drug discovery, drug development, infectious disease research, stem cells. We actually were started with advanced deposits by the Harvard Stem Cell Institute and the Broad Institute to build them a automated cell culture system. And we've gotten pulled into IVF, which is a medical application. And for the first time, we're tangentially related to the FDA. Very much earlier than we expected, about 80% of our sales are to commercial organizations, big pharma, biotechs, and the contract research organizations. So we've raised about $45 million, almost entirely from family offices. The S.A. Lauder family, Peter Thiel are the largest investors, and other family offices, along with various angel groups. We're closing this month on our Series B convertible note. Thank you, Sal Buscemi, for helping us with the raise and continuing to help us with our Series B. But we're just finishing that up. If anybody's interested, you can get in just under the window. And we're going to be raising 25 million early next year, mostly for replacing microscopes, which requires some low-cost engineering, volume manufacturing, a different kind of user interface. So I've been lucky enough to have 7 unicorns, and I have to say that I'm involved at a very early stage, just from the idea through maybe Series C. I'll stay on the board through public offering and acquisition. And none of those unicorns would be there without me, but they definitely would never have been unicorns without a lot of other people. How are we going to make Thrive a unicorn? Well, when I went to a cell culture lab, it was very similar to a company I started, Cytic, where we automated cytology, which reads pap smears. That test was about 75% accurate. And with our instruments, we made it 98% accurate. Pap smears went from every year to every 3 to 5 years. The course of cervical cancer changed because of our instruments. It was an important, ignored problem. Everybody told us you will not be able to change the way doctors and nurses conduct this test. And it was difficult, but we did, and we sold the company for $6 billion. You need to provide a full solution. So often in a business plan, somebody has identified a true problem, and they're fixing it, but the problem is that it's multifactorial. There are other things that have to be solved in order to solve the problem. And many people ignore that. And the more people that ignore something, the more of an opportunity I view it for me. Proprietary patentable products are important. We have 104 patent applications, 56 issued. Basically, somebody has to buy our instruments or buy us. Some of the hardest problems are solved with cross-disciplinary teams. My own background is very eclectic, includes AI and life sciences. We have people who are software engineers and biologists. It's actually very difficult to produce a really good product without having all of those people communicating with each other. And you have to keep your eye on the exit, which I do. So I mentioned you need a full solution. So just having a better microscope isn't enough. There's a lot of other software that's needed. You need to be able to control the environment. Many microscopes, you don't. You take the plate out across multiple environments, put it in a microscope, look at it, take it back, lose all your data. In order to scale, which cell culture and cell-based experiments cannot, you need documentation and setup tools to make people more efficient. You have to acquire enough data and the right data, and you have to do it automatically so the data is comparable across time. This is one of the biggest problems in research. The data that one lab has is not the same data as another lab, even though they're looking at the same cells. And you have to basically put that data somewhere that's accessible and be able to analyze it. Our instruments do all of that. So we have on the market 4 instruments, but 2 in particular, a benchtop single plate version and a automated 50 plate version that images 50 plates 24/7 on whatever schedule that somebody wants. We just introduced a groundbreaking piece of software that accurately counts cells, which sounds like something we should be able to do, but we don't. The industry does, the field does not do that. I'll explain how we do that. And most of our effort right now is on applications. So replacing various assays, analyzing embryos, analyzing stem cells. So this is the Cellus 50. And for the first time, researchers are able to study thousands of organoids, for example, in 50 plates remotely and with metrics. What's really different about our instrument is the imaging. There are many reasons for it, but these are the three most important. We take images at least 100 focal planes, 2 to 50 microns apart. And the researcher determines it. It's different for each application, but no instrument gives you that kind of control. For those of you who have ever looked through a microscope, you might remember that when you adjust the knob, you get information. You look at the top of the cell, the middle of the cell. Well, other people kind of neglected what people like, but we provide that. But it also not only gives them information when they're viewing it. This is vital for deep learning, data mining. We track in our time series, we get back to within 15 microns of every cell. Cells are 200 microns. And we can do that over months. We're never farther off than 15 microns. We just introduced what I mentioned is groundbreaking, which is we're the only company that builds a 3D model, as you can see on the screen, of every single cell, of millions of cells. We use about 15 NVIDIA expensive chips in order to do this. This is a huge amount of data. Most of our customers are generating terabytes per week. They don't know it. That would scare a lot of biologists. My co-founder built the first sequencers with Lee Hood. And those instruments were really the first ones that collected massive amounts of data, and they had to address that problem. Data management is incredibly important. But we also collect about 80 characteristics about each cell: area, perimeter, irregularity, eccentricity, thickness, roughness, so textures. This kind of data set is ideal for drug discovery and is how our customers are mostly using it. Now, I mentioned cell counting. Cell counting is fundamental to many workflows. How many cells do you put back into a person in therapy, interpreting results? But the gold standard is almost 40% inaccurate. That's because everyone is trying to find the outline of the cells. And when cells are getting close together, it's really hard to do that. And also debris looks like cells. But we look at that topographic map that we have of each cell, and that doesn't look like debris, and each cell looks like a cell. And the closer they actually get, the more pronounced their height. And we also find the nucleus. Well, cells have one nucleus, almost always. Everyone's trying to find the outside, we're finding the inside, but these cells are unstained. We never stain anything because we're following the process of live cells. Because of our stacks, we're actually able to find that nucleus. So I mentioned, I want to run through a few of the applications. So why is counting important? Well, in growing stem cells, and iPSCs is a type, I don't know if people are familiar, induced pluripotent stem cells. It's the predominant way that stem cells are grown. You can basically derive stem cells from your skin, from fat tissue. One of the greatest difficulties we have is expanding, growing. The expansion rates are about 5%. So 5% of the colonies that are picked successfully grow. This is one of the biggest problems in scaling this research. But that's because it's done by, gee, that colony looks good. And sometimes there's intuition that's really there. But by using a little bit of data, we have a customer with a very difficult neuronal cell line that went from 5% to 70%. Nobody in the world has ever done that. And in order to have, this is in Japan, this is actually an improved therapy that they weren't able to scale, but they could with a little bit of data. This is organoids. This is my favorite, probably the most important slide. So remember I said we capture all this data on cells, shape, size, texture. We do the same for an organoid. So an organoid is a collection of cells, and the reason it's so powerful is it's closer to the anatomy. This is where really the best research is being done. Rather than just studying individual cells at the bottom of a plate, which is what we've been doing for decades, we can now study the interactions between cells. The problem is that nobody's been able to really image them. Nobody has been able to reduce the variability. But by having all this data, this is a big pharma, they were able to screen thousands of drug combinations on each organoid and make sure that they could stratify the data so that they were looking at similar organoids along texture, size, shape. These kinds of data sets are obviously extremely powerful. And some of our customers have completely reversed their view of what is important in looking at the effects of a drug on a cell. This is another assay. For the first time, they were able to screen 24 different drugs. They never could have done this to see how quickly the cells move in, kind of like wound healing. This is a viral plaque assay. We have the customer who said Thrive will change the world. During COVID, they did not want to touch COVID's SARS-CoV-2 cells again. The way cells are studied is the virus is put on top of cells, and we wait for a hole to form because that shows that they're an active virus, and then we count them. But we can only count them and see them well when we stain them. They wanted to do that without staining, because to stain you have to touch the cells again. And not only on the far right is what it looks like staining. Not only did they not need to stain, they didn't need to wait 8 days. They could see it clearly at day 5. And we have a very large machine learning project to make that within 24 hours. This is a workhorse of infectious disease research. It will speed up our response to pandemics, and it is also a potential diagnostic. It may be boring, but documentation is critical for reproducibility. And right now, documentation is usually a Post-it note on a cell plate or a Sharpie, not the kind of extensive data that we have, so that people can really look back, researchers can look back when an experiment doesn't work and see why.
We got about 5 minutes.
Okay, thank you.
You talk about machine learning. Where's your machine learning team? Are they here in the States or?
Yeah. We have most of them are in Boston, and they make more money than you or I will make ever. We tend to get younger folks and train them because it is so expensive. So, but our first step before machine learning, we're now using machine learning in applications such as to identify that plaque, not wait 8 days until staining, look at it within 24 hours. We needed the data sets first, and we've now built the instruments that can produce the data. So now we're focusing on applications that are using machine learning.
I thought, you may not remember me, but I invested in 2018, so it's been a few years. Why don't you tell us a little bit, please? We did the Series A convert, and I don't know if you're the pricing round for B, but give me an idea of the pricing then and the pricing now in the next round.
Right. So the question is about our pricing. We have not had a price round for 4 years. That valuation was about $42 million. We're going to be doing a Series B early in the year. I don't know exactly what the number is. We've gotten some term sheets at 2.5 times that amount, so $100 million range. We are implementing the strategy of replacing microscopes as a target. This is not a pivot. It was on our roadmap, but we discovered that our customers stopped using their microscopes because they could walk away from the plate, collect more data than they ever would have it accessible in the database. We were going to do that later, but our customers have shown us that there's value in doing that now, and that will allow us to scale faster. But it is it's low-cost engineering and manufacturing and different distribution. So a lot of the Series B, which will be 25 to 30 million, will be for implementing that, which we believe will be the fastest way to scale. That also gets us in the lab. All of our instruments network will continue to sell what we're selling, but this is a great way to get into every lab. And what we want is to be collecting all the data in all cell culture labs. So that's some of the reason behind the Series B. Sure.
Are there not-for-profit institutions, customers that would like to access your platform but can't afford it?
Yes, and we wish we had more money. Part of the use of the Series B is actually for us to build 10 instruments and give them to really bright researchers doing some important things so that we can enable it. They get equipment they wouldn't have gotten, we get papers.
So the question is, with Mission Capital, if it's available to you to produce those instruments to train the people, would that be a useful resource?
That's an interesting idea. The question was, are there nonprofits that want to use our instrument? My answer was yes. There are also schools that want to use it for training in cell culture. And the follow-up question was, would Mission Capital be useful in paying for those instruments? And the answer is yes.
Training.
Right. And to train, we need to retrain everyone, actually, on the way cell culture is done. And programs in schools, we've had a couple of schools that we donated to and used it, and it was fantastic. But we just haven't been able to afford it. So that's a great idea.
So we'll chat.
Okay, that's a great idea. I hadn't thought of that. Thank you.
Thinking about cells, thinking about thriving, and maybe a personal question. So to maintain one's health, longevity of health, what have you learned? What do you find interesting from your work and research?
Much of the work on longevity centers around the cell, and it has been held back because of really the lack of data and the lack of ability until our instrument to actually follow a cell over time. We have customers that are screening thousands of drugs for the first time, not only on individual cells, but on organoids. And I can't share it, but there are, these are mostly big pharma, and they are in the preclinical stage. For the first time, we're told they can actually study aging on cells and are headed, I think we're going to see a number of drugs that will move into phase 1. Unfortunately, that whole process takes a long time. So hang on as long as you can.
So do you personally do anything? Think about this or learn this?
So, we support researchers who are, and we help them with machine learning. I mean, as a young company, we do everything we can to make our customers happy and succeed. And they have had success in using machine learning and neural networks, finding compounds that were not known that are protective of the cell. Sometimes it's reducing inflammation. There are some compounds that are completely surprising. They don't know what the mechanism of action is. So really our role is as providing tools. Our instruments help almost everyone in life sciences do what they're doing better, and particularly on the therapeutics side.
So the valuation now is 100 million, the pre-money?
It hasn't been set, but that's where we're headed.
So we had a lot of thoughts about this, that there was a value add in the instruments and value add in intellectual property and value add in the cloud data. Are you guys leaning one way or the other?
So we would love for our customers to have data in the cloud, because we could help them analyze it, we could aggregate it, but pharma is so conservative about letting any data out. Somebody at Google told me that pharma is the second lowest user of the cloud. The lowest is railroads. This paranoia, it will go away at some point, but it is holding back our ability to have cloud-based tools. But what the pharmaceutical companies are doing is they're creating their own data warehouses, their own data lakes. So it's actually kind of driven them to realize, well, at least we got to collect what we have. And our instrument, we have a big pharma that has it across groups across the world. For the first time, they can collaborate, they have the same data. But some of the things you and I have talked about, cloud-based, are probably going to develop slower.
It's just amazing because I mean, the two nonprofits that that Norm and I work with, are able to get through this whole problem of, I mean, they're collecting data from all these data lakes in big pharma and then aggregating it.
And they're able to get the data from big pharma.
Yeah, they're nonprofits. I mean, so Stand Up to Cancer and Every Cure.
Well, that's interesting. Maybe perhaps working with a nonprofit would give us
I was gonna
access.
say, maybe there's a way to just tie your
Yeah, that's
analytics
interesting.
into that engine,
That's interesting, because we're not the only private company they say this to.
No,
They say everyone.
the original assumption
Right.
is that it's highly valuable and very secretive, but on the other hand, they're just handing it over
Right,
to build these AI interfaces.
interesting. So, perhaps we ally with the nonprofit.
Something, yeah, maybe there's a way to structure it, but, and are you guys working with any kind of licensing or
Yes.
royalty people? or-
Yes, you asked about, so we've had three big pharma, three of the largest. We're not, we believe that most of our competitors, near competitors, are violating or infringing our IP, but we're not litigious, and we're still filing so much. 104, it was 75, I think, at the end of the year. So 25 or so a year, we want to keep filing. But three large pharma came to us because general counsels generally don't want to fool around. And of course, they don't say we're infringing, they just say they want a license, which is great. So we have always said that we expected to do some monetization. We have three significant licenses in development. So that's something where you had kind of encouraged us to look at starting a couple of years ago.
Yeah, I mean, I think it's a great opportunity.
Yeah, we're really pleased with, this will be margin, almost 100% margin revenue.
That's nice.
Yeah,
That's a good number to
I'll
be
take
shooting
that.
for.
I'll take that. Any other questions? I don't know if we have any time.